Okay, let's unpack this. Imagine you're trying to find the specific bit of DNA that makes a plant tall.
Yeah.
Or maybe, like in some of the material we looked at, what influences body weight in mice.
Mm-hmm.
It feels a bit like finding one tiny thread in a giant tangled ball of yarn, doesn't it?
Oh, absolutely. Like a needle in a haystack, for sure.
Right.
And that challenge, well, that's exactly why geneticists need these clever statistical tools to help them, zero in on potential gene locations.
Which brings us neatly to today's deep dive. We're exploring a really powerful statistical method called interval mapping, or IM.
Yeah, and this deep dive is actually built right on the transcript and the class notes from Professor Guilherme de Silva Pereira's statistical genetics
Oh,
course.
great source. That's at the Department of Agronomy at the Federal University of Viçosa.
That's the one. It gives a fantastic look into how these methods, like IM, actually work in practice.
So our mission today is to understand why IM was such a significant step up from the simpler approaches.
Right, and how it helps researchers pinpoint potential gene locations, ideally with more accuracy.
Okay, so before IM was a basic way, single marker analysis, SMA.
Exactly, SMA. And it's just what it sounds like. It looks for a direct link between a trait, like height, and the individual genetic markers you've actually genotyped.
Okay, simple enough. But genomes aren't always perfectly saturated with markers, are they? You always have gaps.
That's a huge issue. You have gaps between markers, and your data might not even be perfect. Maybe some genotypes are missing, or there are errors.
So with SMA, you only see what's happening right at the markers you have data for.
Precisely. You completely miss anything important happening between those markers in the intervals.
And that's the gap, literally, that interval mapping tries to fill.
That's exactly the problem it was designed to solve. It systematically searches within those intervals.
So it goes beyond just the points we measured.
Yes, it's an extension of SMA, but it looks inside the gaps using the information we do have.
Okay, so how does it actually search inside those intervals where there isn't a direct marker? That sounds tricky.
Well, this is where it gets pretty neat, statistically speaking. IM uses the information from the markers on either side of an interval. We call them flanking markers.
Neighbors, basically.
Neighbors, yeah. And it uses them to estimate the probability that a particular gene variant, say the tall version or the short version, the QTL genotype, is present at any given point within that interval.
Ah, so it's inferring. It's calculating the likelihood of a genotype being there, even though we didn't directly test that exact spot.
You got it. You can almost think of these calculated probabilities inside the intervals as creating pseudomarkers.
Pseudomarkers, okay.
We didn't genotype them, but we can statistically estimate the chance of a genotype being there based on recombination, how often genes shuckle between those flanking markers.
And there's a specific statistical framework behind this, something called a mixture model.
That's key, yeah.
Yeah.
IM uses what's called a mixture model. The idea is if there is a gene affecting the trait in that interval, the trait's distribution, like height measurements in the population, won't look like a single smooth curve.
Okay.
It'll look like a mixture of curves, one for each possible genotype at that hidden gene location. The model tries to find where on the map that mixture effect is strongest.
Right, so IM scans along the chromosome, testing spot after spot within these intervals.
Exactly. It moves along the genetic map, maybe every centimorgan or so. And for each hypothetical position it tests, it calculates a statistical score. Often it's a LOD score.
LOD score meaning?
Likelihood of odds. It basically tells you how much more likely your observed data is if there is a QTL at that spot compared to the likelihood if there isn't one.
So a high score means, hey, look here.
Pretty much.
Yeah.
A peak in the LOD score suggests a likely location for a gene affecting the trait. And the mouse data example in Professor Pereira's notes shows this perfectly. The highest LOD score, the strongest signal for the gene influencing body weight, wasn't right on a marker. It was clearly between two markers. SMA would have missed that precise spot.
Ah, interesting. And calculating these scores, are there different ways to do the math?
Yes, the notes mention a couple. There's the expectation-maximization, or EM algorithm, which is more statistically exact but can be computationally slow.
Okay.
And then there's an approximation using linear regression, often called Haley-Knott regression. It's faster.
And does the approximation work well?
Often, yes. The notes actually show examples from both mouse and maze data, where Haley-Knott gave results very, very similar to the more complex methods. So it's a practical alternative.
Okay, so pulling this together, why was interval mapping such a big deal when it came along?
Its power comes from testing potential gene locations directly within those intervals, not just relying on the markers themselves.
Which gives you potentially more precise locations.
Exactly, especially if your genetic map isn't super dense with markers. And generally, it has more statistical power to actually detect a gene compared to SMA.
But you mentioned limitations earlier. It's not perfect, right?
No, definitely not. A really big assumption in the basic IM model is that there's only one gene affecting the trait within any given interval you're testing.
And what if there are two or more close together?
Then things get complicated. The location estimate can be biased, pulled between the real genes. And worse, you can sometimes get ghost QTLs.
Ghosts.
Yeah, false peaks that appear in the analysis, not because there's a single gene there, but because of the combined effect of two or more linked genes that the model mistakes for one.
Okay, tricky. Any other limitations?
The standard IM also doesn't account for interactions between genes at different locations, what we call epistasis. It looks for genes one at a time.
Got it. So to recap, interval mapping was this crucial step forward. It let researchers peer into the spaces between the known markers,
Using probability and statistical models, like that mixture model, to uncover potential hidden genetic influences on traits.
and it's that ability to use the flanking markers, the known information, to infer what's happening in those gaps. It's the key.
It really is. It gets us closer to the full picture because, well, genes don't always cooperate by landing right on top of a convenient marker for us.
Definitely not. Okay, so here's something to think about. Given that standard interval mapping simplifies things, assuming one gene per interval, no interactions, what kind of more sophisticated statistical methods do you think are really needed to properly untangle traits that are controlled by lots of interacting genes scattered all across the genome?
Oh, well, that opens up a whole other area of research, moving beyond single QTL models. Maybe something for another deep dive.